Hi, folks. So we're going to talk about the rest of Chapter 8 today. Um, so we discussed already mitosis in terms of its division among somatic cells in the body. And remember, these are the cells that typically are everything except your sex cells or your gonads. So they would be tissue, nerve cell. They would be, anything from any appendages, any liver, any heart. Those will all undergo mitosis. So when we took a look at essentially cleavage furrow production, what that involved was when the cells split eventually into two, and you could see two resulting daughter cells with identical DNA from its parent cell. In the case of plant cell mitosis, you observe cell wall, um, cell plate formation, which allowed for two daughter cells to be created in the form of identical cells in relation to its parent cells again. But again, it wasn't with cleavage furrow production, it was with cell plate formation. So if we keep taking a look at cytokinesis, we saw this video earlier in class. Let's go into where we stopped off today for lecture. So really what you're looking at is in the lab, when you observe cells, cells essentially attach to the surface. For example, a petri dish. When you culture them, what happens is they continue to divide until they touch one another. Normally, they'll create a sheet of cells. So if you think about transplant surgeries, burn victims who have to have a fragment of skin replaced by a fragment on another part of the body, they'll usually take that from, let's say, a corpse and pull that skin off of him and go ahead and transplant that directly onto the patient that requires coverage of the burns. So you're going to see a thin layer or sheet of cells. You could see it even up to the point of the epidermis, dermis, and hypodermis, all three layers of the skin or integumentary system completely replaced, especially if the poor burn victim is burned all the way down to the bone. You want to try to be able to cover it, so regeneration of everything in terms of capillary structure, blood cells, tissues and adipose tissue can reoccur. So we're talking about cells that continue to divide until they essentially touch one another. Most animal cells will usually divide when they're stimulated by growth factors. So this is very, very important. Growth factors are factors that are inherent in the tissue. So the tissue itself, like the skin, has skin-derived growth factors. They're called epithelial-derived growth factors. We're referring to the epithelial tissue, which covers the skin and lines organs. Because you've got mesothelium, which is inside the body, epithelium, which is above the surface of the skin itself. So, checkpoint. Compared with a culture, the cells in an experimental culture, compared with a control, the cells in an experimental culture are fewer but much larger in size when they cover the dish surface. So again, what's the reasonable hypothesis for this? And again, this stems from those growth factors. There could be one, there could be multiple, there could be reason why that tissue decides to grow larger rates than it normally does inside the body. That's why when we take a look at transplants, we actually have better survival for that patient to recover after secondary or third degree burns. Typically, a third degree patient is the one that does get those transplants and that skin grafting. But essentially, what you'll see is a higher growth of the tissue simply because there's one or more growth factors involved. Okay, so if we keep going along, we'll notice that here we have the cells themselves, which are anchored. I'm going to blow this up to make sure it's good. I feel like the entire thing isn't showing. I hope it's covering the entire screen. Okay. So we see the cells that are actually anchored, and they will be the precursor cells that will start to create a single layer of cells in the first petri dish plate. If we remove even just one of those cells, what you'll notice is the cells will continue to grow and divide to occupy that space. What unfortunately happens though is cancer behaves completely different from that. These metastatic cells will just form on top of each other and just create a clump where thereby it'll be much more difficult to actually remove that cancer. So you'll have to do a biopsy, which essentially is a sampling of the skin or that tissue that you think is either benign or malignant. And then when you send it to the histology, folks, those guys observe the tissue. They're determining whether or not it's cancerous or not. If it's cancerous, they're going to see an observation of a series of cells that just constantly are actively mitotic, and they spread like wildfire. And that's what this sample picture shows below, where they're kind of spreading and growing right on top of each other. So anything in terms of breast cancer, lung cancer, liver cancer, the cells themselves essentially form clumps and they overlap one another. So one thing we can do to actually kind of grow tissue is we can actually try to suspend, sorry, let's try to get to, we can actually try to suspend this tissue or cells in liquid. And so you see this broth of Erlenmeyer flask, you got the cultured cells. If you want to add growth factor to it, you know what will happen. You'll see a rapid mitotic division, as opposed to here, the control, where the cells just essentially fail to divide because they're not adhering nicely to the suspension, or in this case, the actual control isn't a good mechanism for the cells to grow and divide. Here, with the presence of growth factor, you're going to see an effect that's demonstrated among a huge culture, so you can essentially grow tissue in the lab. I talked about in class that you saw the picture of the mouse with a little human ear on the back. We can grow anything on top of cells, because as long as they start to create a first type of ear cell, they will grow into that full ear until it is fully created, and it'll suspend and stop itself. Okay. All right, so we rely on a series of proteins. Proteins are essentially embedded inside the signaling mechanism of the cell. So what happens is there's a critical checkpoint in the cell cycle that'll determine whether the cell will grow through the complete cycle. We talked about checkpoint 1, checkpoint 2, after growth 1 or gap 2 or growth 2. That's kind of flanking the synthesis phase of interphase. So that's a very important social checkpoint, because if that doesn't happen, the cell cycle will undergo arrest and just stop. There's a lot of growth factors that tend to bind, and there's specific receptors even on the surface of the cell. Remember, the plasma membrane was just a fat lipid layer. It's usually necessary for cell division, so these growth factors like GF1, GF2, GF3, GF4, so on and so forth, are specific for that cell in order to create this growth system. So you're going to see this nice ability to continue its overall tissue growth. So, checkpoint. At which of the 3 checkpoints described in this module do the chromosomes exist as duplicated sister chromatids? So remember, after the S phase and interphase, we had the G2 phase? The G2 phase was where we grew just a little bit. So that's one of the 3 checkpoints. The other one is actually in mitosis. So it's going to exist as a duplicated sister chromosome or chromatid in karyokinesis, which is division of the nut. Karyon refers to the nut or nucleus. Kinesis refers to division, so you just got division of the nut. Okay, got to memorize and understand the cell cycle. The cell cycle has 3 subphases of interphase. You got growth, where you can put the chromosomes right on top of it. You got S phase for synthesis, and you got further growth, which is basically allowing for those chromosomes to finish their final growth spur. Then we got kind of the boring phase of mitosis, which is prophase, metaphase, anaphase, and telophase. Other words, karyokinesis. Karyokinesis can or cannot be accompanied by that process called cytokinesis, which is cell division, where the cell just splits into 2. It's going to go again through the cycle, go through mitosis again, split into 4, go again through the cycle of interphase, mitosis again, split into 8, and you guys get the idea. 16, 32, 64, 128, 256, right? So the story is these checkpoints are essentially these stoplights. The growth or G0 phase, where you're originally growing the phase, this is the first growth phase, there's a checkpoint which will essentially stop if the chromosomes do not grow perfectly. After the S phase of interphase and further growth, you get a second checkpoint. So that ensures that the chromosomes are nicely grown before you eventually get into mitosis. Mitosis obviously is splitting the genetic material now into two, and then after that mitosis, you got an M checkpoint to ensure that even mitosis went through perfectly. All right, keep going. Let's take a look at what happens when you add growth factors. That means if we grow it in the lab, we add these growth factors, there's a series of a signal transduction pathway where essentially the growth factor attaches to the receptor, it binds to, let's say, a hormone like a protein. It'll then undergo a signal, it'll transduce and basically control the system of interphase and all the phases of metaphase, right? Prophase, anaphase, and telophase, which are all the phases of mitosis. Either which way, this checkpoint allows you to pass through that, let's say, train system. You know, you open up the barrier, eventually able to go through all subphases of interphase. Okay, if we keep going on, let's keep talking about cancer, just because even in excess, when it forms, we know that there's two ways that can invade other tissue. If it's metastatically growing, basically it's a malignant tumor. And this involves chemoradiation, perhaps the advanced phase of therapy called immunotherapy, which basically means we want to stop their cell division. If you remove the malignant tumor, the cell that's canceling metastatically growing, that doesn't mean that the cancer will disappear. If it's in remission, it may come back, especially in cases of advanced cancers where there isn't continuous therapy as you go along. As I said in class, when you do chemoradiate the body, that cancer, that radiation will stay inside the cells and even the normal cells. So it'll radiate even the healthier cells that are viable and perfectly fine and not metastatically malignant. And they'll get rid of those as well as the normal malignant tumor that's growing and obviously creating havoc in the body. Cancer itself, it can't be obviously, it can't be detected, it can't be diagnosed unless there is a biopsy, which is a graft or a pull of the tissue. So you can take a look at it underneath the slide and observe it with the histology that we observe with tissues. If it's normal, it's called a benign tumor. Now, tumors can exist throughout the body, right? Benign tumors are just accumulations of cells that are not metastatic. They're not malignant. They're not bad for you. So there's many things in the body that can grow with excess, and mitosis can just split the cells over and over and over again, but it doesn't actually create havoc on the body. If one of those cells eventually become malignant, and let's say there's a growth factor or something that triggers that gene to turn on, and the oncogene turns on, then of course it'll turn into a malignant tumor. We don't know why they happen, you guys. There could be genetics, it could be engineering of the body, it could be environmental, it could be no history at all in the family at all. But cancer is just one of those scary things that can happen even to the healthiest and the quote-unquote normal patient that you have. Okay, here's an example of metastatic tissue in the breast. So here we have a series of glands and outstretched tissue, and then we have, unfortunately, a tumor-sized ball. This could be golf ball sized. It could be the size of, smaller, a ping pong table ball, whatever it is. If it's growing in the tissue, the worst part is that you're going to have to worry about the lymphatic tissue behind the breast. So if you don't metastatically cut out that tumor, it will constantly invade the body, right? If it's not found early on with self-checks, with self-exams, you raise your arm, you lay down, and you press against the breast tissue itself. This is kind of if the doctor says right after or before your period. Remember, men get breast cancer as well. So this is one of those things that even if it's a family history in the females of the family, there's a chance that even the male could get it too. So this isn't just a female-only cancer, okay? If it invades then that glandular tissue, it will invade the entire overall breast location. And if it invades the blood vessels and the lymphatic vessels, it will travel to another part of the body. And this is when we're really going to get to, let's say, a type 3, type 4 cancer, where it's metastatically invading the tissues. Okay, So we got to take a look at this. What is really the best cancer treatment? It'll vary from people to people, right? The rates of mortality really vary based upon the diagnosis. Is it type 1, type 1AB? What's the race of the individual? Um, but for example, there is a specific gene in Ashkenazi Jews that gives them a high percentage of a certain disease, right? African Americans usually have sickle cell anemia. Certain races of people will be predisposed just because of a genetic conditions to certain types of anemias or diseases. However, there aren't one race of individuals that are specifically going to get cancer. There's various other factors that align that individual to get it. Stress is one of them. Americans are highly overstressed, underpaid, overworked. Family issues, maybe they can't handle emotional stress very well. They don't take the time out. They're lazy, they're obese. The story could vary, but obviously there's a fraction of Americans, even if you're highly healthy, there isn't much stress on the life. There isn't much environmental or factors that you think that you're doing yourself. You're eating well, you're running, you're taking care of your health. You are, the pinnacle at the peak of a very healthy environment. Even those individuals with all of those great conditions, they can be exposed to cancer threats as well. Okay, so those other factors, they range from person to person. Um, so you got to take this data into account. It may be a randomized location of individuals, or it may be those folks who do have that history of cancer. As I mentioned in class, I have a type 1A cutaneous T-cell lymphoma, which is a type of leukemia. It's a blood cancer. It is a type 1A. So I actually am not getting treatment for it. I don't want to get steroids. I don't want to get anything that'll invade the body. This was a personal choice I did for myself, simply because I know what's in chemoradiation. I know what's in certain types of therapy. There's other things I can do, UV light therapy, that will actually try to change with light maybe the cells that are starting to go awry. But for this point in time, that therapy is like once every month. So based on my insurance and everything. Right now, I'm not getting it, but if I do get good coverage with my insurance, that's another problem that we have in the United States as well. For those folks with good coverage, they do get great therapy. But cancer life-saving drugs are expensive, you guys. There's one drug my father is on with his metastatic cancer that's actually out of pocket $10,000. If he didn't get it covered, if he didn't have a Part 2 insurance besides his Medicare and Medicaid, he'd have to pay for that $10,000 a month. So that's for like 3 injections a month for immunotherapy. Right now, he has to pay $2,000 out of pocket, $8,000 covered by insurance. So it is true. If you are in a low socioeconomic status, I mean, I get paid very little. I get paid essentially like a McDonald's worker. When you take a look at the time and the hours that we put in, we essentially get paid less than McDonald's worker. We make minimum wage. I can't afford insurance. So for several months, I wasn't on insurance. And that's why I couldn't get the treatment I could. Again, I'm 38 years old, young, no history at all. I used to work out religiously almost twice a day. I gained actually 10 pounds the last month just because I haven't been able to hit the gym because I've been super busy. But I'm 138 pounds. And I was 138 pounds before the winter vacation. I'm healthy. I try to, as much as possible, do what I can. But I, of course, got a cancer. So it was just one of those life-changing things that it may improve potentially over time. But I have that history. It's a check mark. It's a red flag on my genetic sequence. So that means whoever I marry, if my boyfriend and I do get married, if he has a history of cancer too, that's going to increase the chances of cancer for our kids. But of course, you shouldn't obviously stop that from you preventing the person who you want to marry. You love him and all of that. But the story is, these decisions in life that we do, you have to take a look at some folks' genetic history, see what's going on, see if they're getting that treatment, and then hopefully they can live long, progressive lives. I told you guys before, everyone with cancer has been diagnosed. I was talking to my oncologist. he told me this very life-changing story. He goes, everyone is getting treated. No one's dying unless you have caught it very late into its type 4 or it's got an advanced stage and there's really nothing you can do. And, you know, you haven't really visited the doctor. So we have to be on top of our medical visits. There could be just one random blood visit. For me, I did a biopsy because I knew that this was spreading over my skin, and I consciously knew the biopsy was going to come back. It took 4 weeks. It took a couple of laboratories to actually say, yeah, this is a lymphoma. And then she told me the news. Obviously, I was devastated, but you know that's what happens. You know, you're young. You know, you haven't had any issues before. You've been very careful, and it just boom, it strikes you. Okay, so why must human cancer research often use an observational method when controlled studies could yield more definitive results? And this is because it's kind of unethical, right? To cause, to force, according to this, cancer causing or cancer curing behaviors on test subjects. So researchers can only observe what behaviors occur during the study, right? It's tough. So human cancer research should use I would argue, a definitive histology, series of tests, whether it's you know, PET, CT scans, ultrasounds of the abdomen and pelvis, CT of the head, neck, and spine. Those are diagnostic tests that will definitively tell you there is a mass. But you need to biopsy the tissue. That means pull the tissue out, look at it underneath the slide, and then that histologist will say, yup, I can see cancer cells. That's it. That patient's got cancer. This is where I pulled it from. If the biopsy isn't done very well, so needle biopsy is not guided, or you can't pull enough of a sample out, it could be a false positive, right? Or it could be a false negative. So doing the right biopsy is the determination of you as a patient as well as the doctor, and ensuring that he's got and she's got the right information to know that you're pulling the right tissue, to assume that you're getting the right cells underneath the scope to say, yes, these are metastatic malignant cells. All right, we could talk about cancer, you guys, all day long, right? It says here, the risk of death from breast cancer 20 years ago, after the system's overall diagnosis, was 96.7% survival rate. There's a 3.5% mortality rate. As you take a look at different age ranges, you could see the age is greater for those folks who the percentages of for those who actually live longer in their early 35. You know, I'm in this category right here. So if I get diagnosed with breast cancer, there's a less percent chance, right, for me to actually fight this thing. As I get older and older, it's going to be harder. And you notice the trends at 60, 69, simply because at this point, too, you're not really, you know, having children, and you're not actively taking care of them either. Between races, breast cancer death rates, unfortunately, among African Americans, Black Americans are higher than Asians, Hispanics, whites, and the majority of the population. This is just one of those things that is a 2018 study done by the authors of the book. Let's take a look at now sexual division. Meiosis 1 and meiosis 2 is basically division of your gametes, your egg and your sperm. Crossing over is that event that takes place in prophase 1 of meiosis 1, which essentially assures that you will allow for higher genetic diversity among the body. Okay, so if we take a look at chromosomes themselves, a couple of things we have to observe is if the body is itself producing cells, then those are autonomic body cells. Those usually consist of 23 pairs of homologous chromosomes, 23 from mom, 23 from dad. One exactly is a human cell,